Okay, I want to continue our discussion today of plant systematics. And as part of that, I want to emphasize again how important it is to go back to our overall life cycle. And again, I can't go through this in as much detail right now, but the overall life cycle, the generalized life cycle, gametophytes producing Antheridia archegonia, sperm ova, sporophyte generation, generating spores, and making sure you have a good foundation in this life cycle because, again, as I talk about other plants, I kind of expect you to know where we are in this generalized life cycle. And so given that, I want to go ahead and talk about our next group of plants. So we previously talked about the bryophytes, which were the mosses, and the mosses you may recall are avascular gametophyte dominant. And so the rest of the plants that I'm going to talk about, which are the plants that we're most familiar with, are the tracheophytes. And so the tracheophytes do possess the vascular tissues, both xylem and phloem, and vascular tissues also are sporophyte dominant, meaning again that the sporophyte is physically larger, it's longer lived, and the gametophyte then will be short-lived and physically smaller. And so we're going to see a transition as we move through the different plants and kind of the evolutionary history of plants that the gametophyte gets smaller and smaller and more simple as we go to more advanced and more recently evolved plants. So again, back in the kingdom Euplantae, the next division I want to briefly mention, an example of a tracheophyte plant, is a group known as the Lycophyta, which are club mosses. And again, they were one of the early types of tracheophytes to evolve, so they are tracheophytes. But what's interesting about this group and the next couple that I'm going to talk about is even though they are structurally more complex now, so we are now seeing true roots, true leaves, and stems, they also still have a primitive life cycle in that they have flagellated sperm, so they require water for fertilization. So again, as plants evolve from the avascular plants to the vascular plants, the vascularization is an important adaptation because now they can exploit drier habitats. They can get water from the soil and take it up to leaves. They structurally are stronger, so they can move up off of the soil. But they are still tied to at least very humid habitats because they need water for fertilization. So again, Lycophyta is an example of this, the so-called club mosses. Again, kind of an obscure group of plants, but you may have seen them in ornamental situations. Another plant like this, which succeeded the Lycophyta in the fossil record, are the Sphenophyta, and they're known as the horsetails. And these are still pretty common. But again, even though this is a plant that you see around a lot, again, what's interesting is that they are, although structurally a little more complex, do require water for fertilization. And so, again, another aspect of requiring water for fertilization is that these are not seed-producing plants. So I think most people have the idea that all plants produce seeds, but they don't all. And we've already talked about mosses, and now the club mosses and horsetails. And so horsetails, again, you may know from ornamental situations, but you see them out in the wild as well. But again, these are plants that at one time in the fossil record were a dominant plant, really are not a dominant plant today. And again, one of their limitations is that they do need water for fertilization if they want to practice sexual reproduction. Okay, now, one of the more extreme versions of this kind of plant that has structural complexity yet a primitive life cycle are the ferns. So again, ferns succeed the horsetails basically in the fossil record, were a dominant plant in the Carboniferous area. And again, tracheophytes that require water for fertilization. So I think most people are familiar with ferns, and again, they're still a fairly successful group of plants today. But again, I think most people just intuitively think that ferns must produce seeds, but they do not. So again, tracheophytes require water for fertilization. And again, part of being a tracheophyte is that they are sporophyte dominant, and that would be true of the club mosses and the horsetails that we just talked about. So again, when you look at a fern, you're looking at the sporophyte. This is a diploid plant. And they do produce gametophytes, but the gametophytes are going to be small and short-lived. So the gametophyte literally in this case will be a separate plant, but it's literally maybe a 1/16 of an inch or maybe as much of a 1/4 of an inch in size, but they're only going to live for a very short period of time and are certainly not the dominant generation here. Okay, so as we look at the life cycle of ferns then, it kind of begins with the sporophyte, and we'll start there anyway. We can start anywhere we want in a life cycle, but of course the sporophyte is a diploid plant, and the sporophyte is going to produce, its job is to produce spores. So it is going to produce sporangia, but the ferns are kind of interesting because they will produce clusters of sporangia on the fern fronds, on the leaflets of the fern frond. And so there's a relationship of terms here and structures that I want you to be familiar with. One is the sorus. So a sorus then would be these clusters of sporangia that we find on the bottom of the leaflets of the fern frond, and the sporangia in turn produce spores. So I'll go to the Elmo here, and so what we can look at here is we're actually looking at one of the leaflets of a fern frond, and so you can see the size based on my fingernail there. But these little dots that you're seeing there are the sori. So these individual dots are sori, and the sori are clusters of sporangia, and of course the sporangia then, that's where meiosis will occur, and haploid spores are produced within those sporangia. So let's look at this, if we were using a microscope to look at this, kind of looking through the surface of a fern leaflet then. So looking through the leaflet, here's the sorus. The little individual circular structures are the sporangia. And on a higher magnification, again, the entire structure is the sorus. Here's the sporangium. The individual circles are sporangia. And then within the sporangia will be the spores. So here's a cross-section looking at a fern leaflet that we sectioned through. And again, here would be the sorus, composed of individual sporangia. And a closer look at the same thing, sorus and individual sporangia. And then within the sporangia, you can see there's some little structures. And if we go up on a high power, then we're looking at an individual sporangium. Here's the sporangium and the stalk of the sporangium. And these are the spores inside. So this is all diploid tissue. The leaflet, roots, everything's diploid tissue, including this tissue here. And when within the sporangia, meiosis occurs, and that's where the haploid spores are generated. And so then the sporangium will rupture and the spores are released. And again, haploid spores. So you may recall then that spores are released in our general life cycle and that spores will grow into gametophytes. So in this case, the gametophytes will be quite small, generally 1/4 of an inch or less, usually kind of heart-shaped like this. And then on the gametophytes, remember the gametophyte plant, it's a plant that produces gametes. Its job is to produce gametes. So in some ferns, these will be separate sexes, sometimes they're combined, but on these gametophytes then we will find antheridia and archegonia. So let's go ahead and again, in the overall life cycle, again, we're now in the gametophyte stage, so we will have gametophytes that produce antheridia and archegonia. Antheridia produce sperm, archegonia produce ova. So looking at an example of a gametophyte from a fern, this would be a fern gametophyte, and this one is producing only antheridia. So each of these little dark structures is an antheridium, and they tend to be spread all over the surface of the gametophyte. And if we look closely, we can see the individual sperm within. And so those will rupture, sperm are released, and again, they will have to start swimming. They're flagellated, and they will look for an ovum within an archegonium. Here is a gametophyte, fern gametophyte that has a cluster of archegonia. And these gametophytes are somewhat heart-shaped, as you can see, and they also essentially kind of fold in the middle. So, you know, they look kind of like a V if you were to look at them head on. And so that allows actually water to aggregate on the surface of the gametophyte. And so, again, if there's sperm in the water, the sperm can swim and they will swim right down the top of these archegonia. So again, remember that the archegonia are like a little vase, and so they can swim down the top and then fertilize the ovum in between, at the base. So we're actually looking right down the top of the little vase-like structure. The sperm swim down, fertilize the egg, which would be in the center here. And then, once that egg is fertilized, then it's no longer an ovum or an egg. It is now a zygote. So it would be a diploid cell. And so that diploid cell then would be fertilized within the archegonium, and then the sporophyte would begin to grow from that zygote. So the zygote will mitotically divide and begin to generate a sporophyte. And so this is what we're seeing here. So this is a gametophyte plant here. Archegonium was right here. It was fertilized at that spot, formed a zygote, and then that zygote began to divide, and this is a baby sporophyte, if you will. So it's starting to produce a rhizome or a root, and it's producing a stem and leaves. So what's going to happen then is that sporophyte grows, that it's literally just going to destroy the gametophyte. It's going to rip the gametophyte apart, and that's the end of the life of the gametophyte. It's done. So again, ferns would be sporophyte dominant because the sporophyte is bigger, longer lived, and in this case actually destroys the gametophyte, which might only live a few days or a few weeks. All right, now, getting back to the Euplantae then, there are also a couple of subkingdoms that are very important, and we're next going to transition then to plants that are not only structurally complex, but have a more complex or more advanced life cycle as well. So these would be the seed-producing plants. So there are two major groups of seed-producing plants, the gymnosperms and the angiosperms. And the gymnosperms produce seeds that are quote-unquote naked, either not protected in any way, or they'll develop within a cone. And the angiosperms produce seeds from flowering plants, and the seeds will develop within a fruit of some type. And so that would be their major characteristic. Now, again, both groups then are seed producers. And what goes along with that is that they use pollen for fertilization. So again, this is a major adaptation. Seed producers use pollen for fertilization, which means then that they no longer need water for fertilization. So they can disseminate pollen by means of wind, or they can exploit pollinators of some type. They might be exploiting insects, they might be exploiting birds like hummingbirds, they may even be exploiting bats to carry pollen from place to place. And we'll talk a little more about that as we get into some of these life cycles. But the point is that this is a major advancement for plants to invade drier habitats. So certainly they have to tap into water in the soil and be able to bring water up through transpiration. But now they no longer need water for sexual reproduction. And so again, that opens up completely new habitats to plants. So before we move ahead then, it's important to know what a seed is, what makes up a seed. So one component of a seed is that it will actually have the embryonic plant. There's a baby plant there, a baby sporophyte, so it's diploid, and that's an important characteristic. There will also be nutritive material as part of the seed called endosperm. And the endosperm will be haploid in gymnosperms, and it will be actually triploid in the angiosperms, or flowering plants, and we'll talk more about that later. Then there's and again, this endosperm then will provide nutrient material to the young embryo to get started. Also, there will be a protective seed coat, which is kind of the maternal tissue. It will also be diploid tissue. And then another aspect of seed production I want you to be aware of is that the seed develops from an oval mass of tissue known as an ovule. So one ovule yields one seed. So each seed is derived from a single ovule, and we'll get back to that as well. Okay, so again, looking at seed structure then, we're going to have the embryo, embryonic plant, the baby plant, sporophyte. We're going to have endosperm, which is nutritive material that will help it get a head start in life, basically, provide it nutrients, help it get up out of the leaf litter or the soil. And then protective seed coats as well, which are derived from, again, kind of the maternal tissue. All right, so first looking at the gymnosperms. So characteristics of the gymnosperms is that they are tracheophytes. So they're vascular plants. They have xylem and phloem. They do not require water for fertilization because they fertilize via pollen. They are sporophyte dominant. So again, when you look at gymnosperms, you're looking at a sporophyte plant, diploid plant. Again, as I mentioned, they fertilize using pollen. That also implies or dictates that they will be seed-producing plants. And here, kind of the key characteristic is that the seeds develop either within cones or they're quote-unquote naked, unprotected. So again, looking at the gymnosperms, gymnosperm division, some examples of gymnosperms include cycads, also called sagosperms. So this would be division Cycadophyta. And again, the cycads kind of look like palms, but they're not true palms or true palm trees because palms don't produce the cone. But here you can see actually that they are producing a cone. They'll always produce what we call a male cone or staminate cone. They don't always produce a female cone. Sometimes they have naked seeds. Another gymnosperm would be the ginkgos. And ginkgos are kind of unusual in that they have kind of a fan-shaped leaf, and they produce naked seeds, usually in pairs. A lot of times those seeds drop and ferment and can be quite smelly if you have these growing around your house. But ginkgos are common ornamental plants, and ginkgos are also interesting because they're separate sexes. There are male trees and female trees. The nitophytes or Nitophyta would be another group which are kind of unusual. We don't see a lot of them in the United States. In the United States, there's one called the Phaedra, so that would be one that's common in this country. But they're a fairly bizarre group, but again, they tend to produce either naked seeds or seeds within cones. And then the one that we'll spend most of our time talking about are the conifers. And so conifers, of course, include things like, firs and pines, and redwood would also be an example. Also, different types of junipers would be examples. These are all examples of conifers, which are a type of gymnosperm because the seeds develop within cones. So we're going to look at some detail then at the life cycle of a conifer. And so when we look at a pine tree, so when we look at a conifer, when we look at a pine tree, we're looking at the sporophyte plant. And again, keep in mind that that means that the plant will produce spores. Now, they also produce cones. And so they will produce both male and female cones in most cases. So most plants will produce both male and female cones, although they do have some mechanisms to prevent self-fertilization. So we're going to look first at the male cone. And so the male cones on a tree will typically be very small, and this is referred to as a staminate cone. And so as we look at the structure of the cone, and I'll come back to this in a second, is that the staminate cone is a reference to a male cone. Stamen is a reference to maleness. And also the prefix micro, when we talk about plants, also refers to males. It actually just means small, but it's a reference to males because they will have a small gamete typically. So the staminate cone is sometimes called the microstrobilus, and so this term strobilus is also a reference to a cone. And the strobilus is composed of scales, basically modified leaves that are called sporophylls. So this would constitute a sporophyll, a leaf then that makes spores. So the suffix "phyll" is a reference to leaf, spore, a spore-producing leaf. So again, the sporophyll then would be a spore-producing leaf. And the term microsporangium means a microspore or a tiny spore. And so in this case, that's a reference to the pollen grains. So again, the staminate cone is the entire cone. It's essentially a bud, a leaf bud. And so this is the modified leaf, the microsporophyll. Within the microsporophyll is the microsporangium. So this is where the spores are going to be produced. And then the spores themselves are the pollen grains. So the microsporangia then are going to produce spores. The spores then are the immature pollen grains. And you may recall that spores are just one cell. And typically they're disseminated, and that's going to be the case here. But the spores then are the immature pollen. When the spores undergo a single division, it's no longer a spore. If it undergoes a single mitotic division, it's now a gametophyte, a very small gametophyte, but a gametophyte nonetheless. So when the pollen grains, which have a single nucleus called microspores, undergo a single division, that forms a microgametophyte. And these are now the mature pollen grains. So microgametophytes then, again, sporophyll is the individual scale. The sporangium is this large oval structure. Within that, the microspores are produced, which are the immature pollen grains. And then when they undergo a single division to produce two nuclei, those are referred to as microgametophytes. So this would be a collection of both microspores and microgametophytes. And then eventually this ruptures, and then the pollen grains can be disseminated. So what I want to actually show you, though, before I move ahead, is the actual size then of one of these male cones. So again, we were looking at that with a microscope, but if I can just put this over here on the Elmo for a second. Anyway, I'll just put my finger here. So again, that gives you an idea of the size of a male cone on a tree. So again, compared to my fingernail, we're talking about small structures. So these tend to be found in clusters of cones that are known as catkins. Okay, so again, as we consider the difference between a microspore and a microgametophyte then, the microspores would be immature pollen grains. They're not fully mature yet, and they only have a single nucleus. Microgametophytes are also pollen grains, but these are the mature pollen grains. They will have two nuclei. One of those nuclei is called a tube nucleus, and the other is called a generative nucleus. And it's the generative nucleus that's actually going to be responsible for producing sperm that can fertilize an egg. So again, as we look at these microsporophylls, the microsporangia, and then the microspores, if we go up on higher power, what we can see here is that some of these have two nuclei, tube nucleus and generative nucleus, and some only have a single nucleus. So this would be a microspore. This would be a microgametophyte because it has the two nuclei. Also notice that in the conifers, the pollen grains have wing-like structures associated with them, and so this is telling us that these are wind-pollinated. So the conifers wind-pollinate. So the sporangia rupture, the pollen grains are released, and wind will carry those pollen grains away. Okay, now, while that's going on in the male cone, the female cone is also developing. And again, it's basically the same idea, except that in terms of relationships of structures, in that we are looking at sporophylls, but in the female, we refer to these as megasporophylls. And at the base of every sporophyll, there will be an oval mass of tissue known as an ovule. Okay? So, which fits actually, for once, the term actually describes what it is. So it's an oval mass of tissue, and this would be sporophyte tissue. And within that sporophyte tissue, a single large spore will develop, known as a megaspore. And then that megaspore will germinate and produce a small megagametophyte. So first the megaspore is within the ovule, and then the megaspore mitotically divides to produce a megagametophyte within the ovule. So this is kind of a divergence from what we've talked about previously, in that every other plant we've talked about to this point, when they produce spores, they basically disseminate the spores. Or in the case of the pollen grain, the microspores form microgametophytes, and those are disseminated and released. But in this case now, within the female cones, within the megastrobilus, the female has this ovule, and she's going to produce a spore within that ovule, and then that spore is going to develop into a tiny gametophyte, but she's going to hang on to that gametophyte. She's not going to release it to the outside. She's going to keep it. And then she can provide the nutrients and protect it within that cone. Okay, so this is a different kind of strategy, if you will, evolutionarily. So the ovulate cone then is composed of megasporophylls. So again, here's our ovulate cone. And the megasporophylls then would be the sections of that cone. So each section or scale of the cone is a megasporophyll. And then an ovule will form at the base of the megasporophyll, as we've talked about. And then an ovule within that, or a megaspore, and a megaspore within the ovule, as I've already talked about. And then a megagametophyte within that ovule as well. So again, just showing you those terms, but we've already discussed that process. And then the last step then is that megagametophyte, among its many cells, will produce an ovum. So there will be a single ovum within each megagametophyte. And unfortunately, I don't have a picture to show you that, but the point is, spore mitotically divides into megagametophyte. Megagametophyte will produce an ovum within the ovule. And the ovule is at the base of each scale of the cone. So, essentially looking at this isn't a fully developed cone, but if we can go back to the Elmo again, just to show you what we're talking about here. I think everybody's familiar with pine cones when they're fully mature, but these female cones when they're immature are quite small. So again, if you can see the size of my fingernail, the female cone starts off being extremely small. And so at this stage then, what happens is that pollen grains are disseminated, they stick in the sap of the cone, get pulled into the interior of the cone, and here's our ovule. The gametophyte is developing, and a single ovum will develop within this gametophyte. So pollen grains are pulled in here, but the pollen grain needs to get to the ovum, which is going to be within this gametophyte. And so what's going to have to happen is that the pollen grain will have to elongate. It grows what we call a pollen tube. So when carries the pollen to the ovulate cone or the megastrobilus, as it dries, it pulls the pollen grains into the interior of the cone, but it still has to get to where the ovum is. So the pollen grains then grow what we call pollen tubes. So we have a tube nucleus and a generative nucleus. And so literally the pollen grain will begin to elongate. And the tube nucleus basically drives the tube. So it elongates the tube, determines where the tube is going to go, it picks up chemical signals from the egg and tries to find the egg. And the generative nucleus will undergo a single mitotic division to form 2 sperm nuclei. So again, the pollen grain will elongate, enter the megagametophyte, which is the female gametophyte, and it will attempt to fertilize the egg, or will fertilize the egg. And so again, that's all happening within the female cone. So when we look at the fate then of these nuclei, the tube nucleus basically disintegrates and dies. One of the sperm nuclei will also die, and then the last sperm nucleus will fuse with the ovum. Those nuclei will fuse, producing a zygote, a diploid cell, and then that zygote will begin to divide and produce an embryonic cell through mitosis. Now, that's one component of the seed then. So the embryo then will continue to develop at the base of that scale within the ovule. Now, you may recall then that ovule also has other haploid cells as part of it. That gametophyte has other haploid cells. So the zygote then will mitotically divide and grow into the embryo. The remainder of the gametophyte, which is haploid, will then form the endosperm of the seed. So conifers and other gymnosperms then will have a haploid endosperm. And then the surrounding diploid ovule tissue will develop into the seed coat of the seed. And so then that seed then will be found and usually there's 2 ovules and 2 seeds at the base of each scale of the cone. And so again, what was one ovule will then eventually develop into 1 seed. So there are normally 2 ovules at the base of each scale of a female cone, each megasporophyll of a female cone. And so eventually then that cone enlarges and gets much bigger and dries out. And at that point then the sporophylls of the cone will open up and then the seeds can drop out. So to get from this tiny stage of the female cone being green and sappy to this dried out stage of the cone is usually a 2 or sometimes 3-year process. So normally what happens in the first year is that the male and female cones begin to grow. The pollen grains are produced, the pollen grains are disseminated. They will stick to the sap in the female cone and be drawn into the female cone. So that's usually what happens in the first year. And then in the second year, and fertilization may occur in that first year, but it depends on the species. Then in that second year, fertilization occurs, the seed develops, the cone continues to grow, and in many cases it may not open up in that second year, and it may actually be the third year before the cone actually opens up, and then the seeds can be disseminated. So the seed then is a nice evolutionary adaptation because again, it gives the seed, the embryo a head start. It now has nutrients that can be delivered to the embryo. It can grow up out of the leaf litter or soil. The seed also helps in dissemination because we animals then, of course, we're looking for food all the time. And the endosperm of a seed is highly nutritious. The embryo is highly nutritious as well. And so we will gather seeds, hide seeds if we're a squirrel or a bird. We may forget where those seeds are, and then they have a chance to germinate out of the canopy, away from the canopy of the parent tree. Or they may pass through our digestive system and still be viable. We may not have destroyed them in our digestive process. So in addition to just helping with getting the embryo started, it also helps in dissemination of the seeds and just physically distribute the organism. Okay, that's it for today. I'll see you next time.